Our last speaker is Ellen Zhong. Ellen is currently a fourth-year PhD student at MIT Computer Science and Artificial Intelligence Lab, working with Bonnie Berger and Joey Davis. Prior to MIT, she was a research associate at the show research, where she worked on algorithms and infrastructure for performing free energy calculation via molecular dynamic simulation. Ellen is a recipient of an NSF Graduate Student Research Fellowship. Ellen's research focuses on developing neural methods for 3D reconstruction for protein structure from cryo-EM images. And she will tell us about the recent advances in this software. Thank you.
Okay. Yep. Okay. Thank you so much for the introduction and for the opportunity to speak. So I'll be talking about CryoDRGN and its use for heterogeneous reconstruction. As a quick outline of this talk, first I'll be giving a background overview of CryoDRGN, in particular, why we use a neural representation for the structure. Most of this talk will focus on applying CryoDRGN on real cryo-EM datasets deposited on Empire, highlighting some of the results from our recent paper and touching on some of the more practical aspects of using the tool. And then increasingly, as we've been using CryoDRGN, it's raised new questions in how do we want to analyze the resulting ensemble of structures. So I wanted to talk about some new directions in both interactive and automated ways of inspecting this learned distribution of cryo-EM density maps. And then finally, I wanted to share some quick updates on fully unsupervised or ab initio heterogeneous reconstruction with CryoDRGN. So as we all know, all essential biological processes are carried out by proteins and protein complexes. And while we often derive mechanistic insight from a static three-dimensional structure, in reality, these macromolecular machines are undergoing large structural changes in order to perform their function. So what we'd really like is to be able to image and study their distribution of conformations and be able to relate that to how these protein complexes work. Unfortunately, techniques to study molecular motions are rather limited. So my background is in molecular dynamics simulations. This on the right is an MD simulation of the SARS-CoV-2 spike protein. And while this gives you a very detailed in silico microscope into the motions and dynamics of these protein complexes, there are a lot of challenges associated with the computational cost and the accuracy of the underlying models, which is why I'm really excited for the potential of single particle cryo-EM at uncovering these molecular motions from real experimental data. So the crux in structure determination, one of the cruxes in structure determination with cryo-EM is the computational task of 3D reconstruction. So here we have a data set of picked particle images, the typical number being in the hundreds of thousands or millions, and we want to reconstruct the 3D cryo-EM density that we model as generating these images. There are a number of challenges associated with this task. I'm sure many of you guys are familiar with these already. For example, the unknown poses. So we don't know the viewing direction of each of these images, each of these particles within the image. And the one that we're interested in is the so-called heterogeneity problem, which is where we break the standard assumption in homogeneous reconstruction that each of these particles is an identical static structure. So this so-called heterogeneity problem, I think, presents a major opportunity in cryo-EM because each image contains a unique molecule, potentially in a different conformational state. We actually get the experimental data needed to resolve the different, maybe functionally relevant conformations of these biomolecular complexes. And this is in contrast to crystallography, where we've experimentally rigidified every copy of the molecule into an identical conformation in the crystalline array. Unfortunately, you can think how this heterogeneity affects reconstruction, how it makes it a much harder problem. So other than getting rid of as much of the heterogeneous data as possible, the standard approach is multi-class reconstruction or 3D classification or heterogeneous refinement, which is where we model the heterogeneity in our data set as originating from a discrete mixture model of K independent structures. So this might work well if you have only a few discrete states, conformational states in your sample of interest. However, you don't really know that a priori what the conformations of your molecular complex will possess, and you have pre-specify K. And so in general, the identification and analysis of heterogeneity especially continuous forms, is an open problem in cryo reconstruction. So our goal is, can we leverage the expressiveness of neural networks in heterogeneous cryo reconstruction? There's been a lot of advances recently in applying like complex latent variable models in deep learning to extremely large heterogeneous data sets. So we wanted to take some of those ideas and apply it to heterogeneous cryo reconstruction. So our method CrowdDRGN stands for Deep Reconstructing Generative Networks. The goal is to learn a continuous low-dimensional representation or low-dimensional distribution of structures from 2D cryo image data. So there are a couple of aspects of this method. The first is our coordinate-based neural representation of structure. The second is our overall architecture, which uses an encoder-decoder architecture based on the VAE or variational autoencoder. And then the third is how we do inference of this model. So we have exact inference for pose in the ab initio case, and variational inference for heterogeneity, or this latent variable model of heterogeneity. If you're interested in learning more, you can check out our manuscripts and our open source software, which is available at this link here. So just as a quick overview of some of the underlying machine learning methods, the autoencoder briefly is, I think of as a nonlinear dimensionality reduction technique. So if we have an input image, which is an n by n matrix, also n squared by 1 vector, we can encode it using a neural network into a compressed lower dimensional representation, typically denoted as Z. And then we have a decoder neural network that learns the inverse. So it learns a reconstruction from this compressed representation to the input image, or ideally to the input image. We can compare this reconstruction to the input image, compute the error, and use the error to update the weights of the neural network. And so ideally through many rounds of the data set, we'll learn some useful representation, lower dimensional representation that we can then use for downstream tasks. The VAE, at a very high level, I won't really be going into the details, extends the autoencoder as inference of a probabilistic model. I think of it as a regularized autoencoder. So the objective function for training, instead of purely being the reconstruction error between the input image and the reconstructed image, also contains a regularization term on the latent space. So the key idea in CrowdDRGN is our neural representation of volume. So in traditional voxel-based reconstruction methods, the volume or the density map is represented as discrete points on a three-dimensional voxel array. So the goal of reconstruction is to fit the value of the density map at each of these different voxels. In CrowdDRGN, we use a neural network to approximate the continuous function that maps from 3D Cartesian coordinates to the cryo density. So a neural network takes these XYZ coordinates, and then we train it to produce the electron cryo density at this location in positional space. So we parameterize this neural volume as a neural network instead of a voxel array. And I should say, after we train this neural network, in order to visualize the density map in something like Chimera, we would evaluate our neural network on a three-dimensional grid of Cartesian coordinates, fill in the density map, save it as an MRC file, and then you can visualize it in Chimera. There's an additional detail, which is that instead of directly inputting our three-dimensional Cartesian coordinates, we featureize this using a positional encoding function consisting of sinusoids of varying frequency, and this helps with the training and representation capacity of this neural network. And I've been very excited to see this type of architecture take off in computer vision, where they're interested in reconstructing natural volumes or three-dimensional scenes. And so I highly encourage you to check out neural radiance fields if you're interested in learning more about this type of architecture. So why do we want to use this neural network representation for volume? The main reason is to model heterogeneity. So in the traditional 3D classification approach, the goal of reconstruction is to fit k separate voxel arrays from the data set. In CryoDRGN, we simply extend the input dimension of our neural network to include a latent variable vector. And so this vector you can think of as giving us additional degrees of freedom for modeling heterogeneity. If we're one-dimensional, you can think of it as parameterizing location on some reaction coordinate. In general, it might be higher dimensional. So the goal of reconstruction is to now learn this generative model from a continuous latent space that describes the heterogeneity of our volumes or our protein complexes to the three-dimensional density maps. So our overall architecture has this image encoder volume decoder architecture. So this volume decoder is the coordinate-based neural network that I just mentioned previously. To train these networks, given an input image, we encode it into our latent space. So we have some latent code, some like per particle estimate for this input image's heterogeneity. And then if we know the orientation or the pose of this particular image, that determines a coordinate slice. So that's based on the Fourier slice theorem, or how we relate the 2D images to 3D volumes. So we can take this pixel location, this Cartesian coordinate, featureize it with a positional encoding function, concatenate it to the latent code, and then reconstruct this value of the density map at this location in positional space and in latent space, compare that to the input image for the whole slice, compute the error, and use that to update our neural network weights. So the overall VAE objective seeks to maximize the probability of the data set. It's trained from random initialization and is optimized with stochastic gradient descent. So that's the training of these neural networks. Once training has converged, then we have this image encoder. We can take the images in our data set and encode it into our latent space. So this is some lower dimensional space that we can then view the empirical data distribution and do whatever inferences we want on the learned distribution. We can also then use our decoder neural network to generate volumes at different values of our latent variable. So to interactively explore the learned ensemble by generating representative samples. We can also generate continuous trajectories given end states of interest, because we have this powerful generative model that can connect, that can generate new structures given values of our latent variable. And because this latent space representation is actually composed of the representations for the different images, we can actually segment the latent space based on maybe particular features or clusters that we've observed. And if you're not comfortable with the neural representation, you can do reconstruction or refinement using traditional tools. Or if a particular region of the data set has a lot of impurities, you can filter the data set based on this latent space segmentation. So just to summarize, given an imaging single particle cryo-EM imaging data set, we train 2 neural networks jointly to optimize the encoding and decoding of some lower dimensional latent space describing the data set's heterogeneity. There are 2 training regimes. I haven't talked about ab initio heterogeneous reconstruction, where the poses are estimated on the fly. The version that's available on GitHub uses pre-existing estimates of pose. So this requires running a consensus reconstruction on your imaging data set to pose the images before using CryoDRGN to fit the residual heterogeneity. In terms of how do you use the tool, so CryoDRGN is a deep learning software framework written in PyTorch. You'll need GPUs, but this is pretty typical for most standard image processing setups. In terms of input or data requirements, you'll need extracted particles, high quality pose estimates, as well as accurate CTF parameters. Hopefully the software is written in a pretty user-friendly way. There's a couple of executables just to pre-process your inputs and then train your model, as well as a couple of automated steps for running the analysis and generating movies. And there's also a tutorial now available that is linked on the GitHub. So hopefully this will be helpful in like really running through what are the inputs and outputs of each of these steps and what you can expect. And so just as a roadmap for the rest of the talk, that was how CrowdDRGN works. Next, I'm going to talk about some of the results applying this on real data sets. So the first data set we're considering is the Plasmodium falciparum ADS ribosome deposited on Empire. This is about 100,000 particles at a fairly large box size. And the first thing we were just interested is evaluating whether the CryoDRGN neural representation of structure is comparable to the voxel-based representation. So here we just trained the CryoDRGN decoder with no latent variable input on the images from this data set. On the right is the voxel-based reconstruction, I believe using CryoSpark, and on the left is the result from CryoDRGN. So they match up pretty well. If you squint a little bit, the resolution of the neural representation is slightly lower than the voxel-based representation. And part of this is because one way to think about it is in terms of the representation capacity of CryoDRGN neural networks. So oftentimes I get questions like how much, like what are the motions that you can detect? What are the achievable resolutions? This is all affected by many different aspects, including just the latent variable dimension, the specific architecture, which is what I wanted to highlight here. So the number of layers and the width of the layers of the neural network changes the number of parameters in the model, and then that affects the achievable resolution of the density maps. I should also say that's definitely bounded by the input data. So what we see here is that the largest neural networks correlate to this is the map-to-map FSC, the largest neural networks correlate with the voxel-based reconstruction at higher frequencies. And another way to think about it, this is more of a rule of thumb, definitely it's not directly comparable, but you can think about the number of parameters in the different representations. So in this CryoDRAGN neural network-based representation of volume, the largest networks that we've tried have around 11 million parameters. And if you think about the number of parameters or voxels in the traditional voxel array-based representation, the 360 cubed box has around 46 million parameters. So one reason we picked this data set to analyze is that it's been analyzed with other methods for heterogeneity analysis. So there's multi-body refinement in RelyOn, which models the heterogeneity as originating from separate rigid bodies. So this gives you a really nice structural prior if you know that your system can be decomposed in terms of different rigid body motions. And then there's also CryoSpark 3D variability analysis that models the heterogeneity in your data set as originating from linear interpolations between eigenvolumes. So it fits a linear subspace model to your data set. And so in both of these approaches, they were able to see kind of motion of the small subunit of the ADS ribosome relative to large subunit. And so this is what we get with CryoDRAGN. This particular data set, so we've trained a 10-dimensional latent variable model. On the left, I'm showing, so each of these data points is a particular particle image embedded into the latent space, and then we're visualizing it on this 2-dimensional zoom window using principal component analysis. So on the right, I'm showing 20 sampled structures at these different points in the latent space. We see motion of this head group of the small subunit, lots of other elements that appear and disappear. And then one of these structures is the rotation of this small subunit relative to the large. So we can look at that at a little bit more detail. So this is the direct comparison of those two structures. There's this inter-subunit rotation, but there's also the concomitant disappearance of some of these protein and RNA helices, as well as the motion of this entire RNA stalk. So we can validate that this is not just some hallucination of the neural network. So if we look at the latent space representation, this state is kind of separated along principal component 1 and in the UMAP representation, or visualization. So we can take these particles in this cluster, which is about 5,000, do a homogeneous refinement, and then we see the exact same differences between the CryoDRAGN volume and then this rotated state. So that was a relatively homogeneous data set. Next, we were interested in evaluating CryoDRAGN on a very heterogeneous data set. So this particular data set is of the assembly of the bacterial large ribosomal subunit done by my advisor Joey during his postdoctoral work. It has 131,000 cryo images, and through many rounds of expert-guided 3D classification, they were able to identify 4 major states of the assembly states of this ribosome, as well as 13 minor states through further rounds of hierarchical classification. So we train a CryoDRAGN model on this data set, and this is what we get. Again, it's a 10-dimensional latent variable model, and we've visualized it in 2 dimensions using PCA on the left and with UMAP, which is a nonlinear dimensionality reduction technique on the right. And we can already see that there are clusters of the data set in the learned latent space. Because we have the 3D classification labels, we can color each of these particles by which class it was clustered into during the 3D classification. And these clusters that we see in CryoDRAGN align with the clusters from the 3D classification. And if we generate structures at the centers of these clusters, we get the assembly states that were previously published. So the B class is the least mature that only has the base, the back of this large ribosomal subunit. And then the E class is the most mature that has the central protuberance and L1 stalk. If we compare the 3 classification labels of the minor states, we see that there are variations within these large clusters that correspond to the minor states. And then just to show a couple more examples of the density maps, this cluster out here is actually, so this corresponds to class A in the published classification, which is the 70S ribosome, and that was an impurity during sample preparation. And class E, so I'm showing 5 samples from these different centers of the minor assembly states. E1, we have the least mature. E2, we've built in this L1 stalk. E3, there's this shoulder. And E4, we've docked this helix. And E5 is the most mature form. So all of this comes out all automatically from just one training run of CryoDRGN. We also noticed that there is this strip of purple particles right next to the green class. And when we generated a structure from this region, we got this density map that resembled the class C major state of the ribosome, as well as, but it had this RNA helix that was docked that was previously only associated with the class E, or the most mature assembly states. So we can take the particles from this strip, there's about 1,000 images, and do a homogeneous refinement with traditional tools, and we were able to reproduce the structure that resembles the class E, but has this RNA helix docked. And then lastly, I wanted to show CryoDRGN on a data set with significant continuous motions. So this is the data set of the precallic spliceosome, also on Empire. And what I'm showing here are the traversals. We train a CryoDRGN model, and then these trajectories are generated from traversing the principal components of the latent space. So PC1 had this like top region, the SF3B subcomplex and the helicase subcomplex clamping down. PC2 had them moving in opposition. And in general, this gives you a very nice sense of the overall degree of heterogeneity in the system, although the caveat is that interpolation along these principal components can produce non-physical motions. For example, if you have compositional heterogeneity, there's kind of this data model mismatch where if the data set has compositional changes, but we fit a continuous model, when you do the interpolation between your compositionally distinct states, you're going to generate non-physical motion or non-physical structures. So in general, you have to be careful when your data set is not supported along this interpolation path, which motivated this graph traversal algorithm for generating structures and trajectories with CryoDRGN. So this is a series of density maps generated by traversing the K nearest neighbor graph constructed from our learned latent space. And to me, this really represents like a super cool, plausible trajectory of this macromolecular machine in motion. You can kind of get a sense of what it might look like from single particle cryo-EM data. So finally, I've shown a couple of different vignettes on applying CryoDRGN on data sets with very different types of heterogeneity. One key question that we've been thinking about in terms of how to make this software as useful as possible is how do we analyze the resulting ensemble of structures? So Crowddrag and Analyze is the executable in the software package. It runs a series of standard analyses, so visualization of the latent space with PCA and with UMAP, generating representative samples, by default 20, you can do more or less, as well as generating trajectories along the principal components of the latent space. But because these different data sets have very diverse sources of heterogeneity, we also wanted to provide an interactive method of exploring the data set and exploring the volume distributions. So we have a Jupyter Notebook, which is a web-based application for exploratory data analysis, which uses Python, but hopefully the template code is all set up, so you really just have to execute the different cells. And this lets you visualize the latent space, plot it in different ways, visualize the raw image data, and interactively generate new volumes. So just to give you an idea of that, this is kind of a screenshot of a part of the interactive selection feature. One thing that we've noticed is that oftentimes a lot of data sets have a lot of non-structural imaging variability. So for example, junk particles or ice artifacts or these edge artifacts. And this we've seen can often interfere with Crowddrag and representation learning. So you can think about like how these edge artifacts, if this is in the data set, the model thinks the model is required to model this heterogeneity using the latent variable. And so if there's a lot of junk particles, then a lot of the representation capacity might be used to model like weird things on the side of the box instead of the underlying structural variation. So we want to remove as many junk particles as possible. There's this interactive lasso tool that allows you to do that. You can also look at kind of the outliers of the latent space distribution and see images that have, so this is just a sampling, random sampling of images with on the outliers of this distribution. You can see images that have hot pixels or some other artifacts. And this particular data set is the SARS-CoV-2 spike protein. Without removing these outliers, we weren't able to see the underlying structural variability of this data set. The model was too busy modeling some of these junk particles, but after filtering out these kind of junk artifacts, we were able to visualize the underlying conformational changes of the spike protein of the receptor binding domain as it transitions between RBD up and RBD down. And then that was more of an interactive approach. I also wanted to briefly touch on some directions in more automated analyses of the learned ensemble. I won't be, in the interest of time, I won't be going into the details of this data set, but this was done, the analysis is done in collaboration with Sue Fei, who's a postdoc in Bob Sarah's lab at MIT. We trained a cryoDRGN model. This was the latent space we obtained. We were able to see the transition between the intermediate recognition complex, which was the original structures in the publication, two original structures from 3D classification. But we also saw lots of other structures, including some of like additional density for the substrate. So the question is, how do we want to analyze this distribution of structures? We can do it in a more interactive or manual way. So this was annotated by Sue, but longer term, we're thinking of ways of more automated ways of extracting distinct states, identifying rare states, for example, and also visualizing the data set in more interpretable spaces. So the UMAP, the latent space of CryoDRGN, is a relatively arbitrary representation. Can we instead have interpretable reaction coordinates for better interpreting the data set distribution? And then lastly, I wanted to talk about ab initio reconstruction with CryoDRGN. We've been making a lot of improvements to the algorithms and to the compute speed of ab initio reconstruction. And this is just showing on the left CryoDRGN with consensus poses from a traditional refinement, and then the ab initio CryoDRGN on the learned ensemble from ab initio CryoDRGN on the right. And so this is preliminary, but very exciting. And we haven't released this yet, but we've seen lots of promising results in ab initio reconstruction, especially ab initio of heterogeneous data sets. And so our overall goal is to really broaden the scope of cryo-EM to these data sets, or heterogeneity analysis to these data sets for which you can't obtain consensus reconstructions. And optimistically, this would translate to additional classes of heterogeneous biomolecular complexes. So in conclusion, I've talked about CryoDRGN, which is a method for reconstructing continuous distributions of protein structure from single particle cryo-EM data. I think one of the most powerful aspects of this method is that the neural representation of structure of heterogeneity provides a very general framework for modeling heterogeneity. So we've been able to reconstruct compositional and conformational changes, discover new, particularly rare states, and filter the data set from junk particles. And hopefully with this method and with many other methods applied to single particle cryo-EM, we can uncover new structures and molecular motions. And with that, I would like to thank you for listening, and thank you to my advisors, Joey Davis and Bonnie Berger in particular. Tristan Beppler is a former member of the Berger Lab who was instrumental in a lot of the early neuromodeling. Adam Lair, who I've been collaborating with on the ab initio reconstruction, and a number of members of the Davis and Berger Labs, as well as our collaborators, Sue and Bob. So thank you. I'm happy to take any questions.
Thank you, Hélène, for an amazing talk. You have lots of questions in the Q&A. I don't know if we have time to go through all of them, but we'll start with a few. So Yuri is asking, how do you deal with the variation caused by the focus variation in the data?
So ideally, that's modeled by the CTF. So so in the ideal case, your CTF parameters are accurate, and then when you do the reconstruction of the slice, you apply the CTF, and the forward model of the image formation perfectly matches the data generating model if your parameters are accurate. So that defocus variation will be kind of taken care of automatically. If the CTF parameters aren't accurate, that's where you have issues, where now there's this mismatch between what you're modeling the image formation process as and what your data set is actually. And so then you can run into weird things coming out of your reconstruction as the model tries to compensate for that mismatch. But ideally, if the parameters are accurate, that'll be automatically taken care of.
Okay, thank you. Donovan is asking, if initial pose estimation of the particles is poor, does CryoDRGN still find useful features?
Excuse me. So one thing we've seen is that if the poses are poor, if there is some subset of the data set where the poses are accurate, then maybe you'll still be able to resolve those motions. And it also depends on like the degree to which the poses are inaccurate. But there definitely is the case of garbage in, garbage out. If the poses are very incorrect, then you're not going to be able to model anything useful out of it. So that's definitely motivated the ab initio direction for CryoDRGN. The other thing that I do want to say that I have kind of messed up some training runs where my poses are completely randomized by accident, and it still picks up impurities in the data set. So that's in one case in which having bad poses is fine. And I think that's because the images for some of these impurities, like ice artifacts or these edge artifacts, are so different from the like perfect particles that still gets picked up. So that was kind of an interesting little finding.
Okay. From Lugan, are there any plans to adapt CryoDRGN to subtomograms? If so, would there be similar resolution requirements, namely the visibility of secondary structure?
Mm-hmm. So there are definitely, so it would be really easy to apply CryoDRGN to subtomograms if, I remember in Alberto's talk yesterday, if you're able to extract the slices from the tomograms. So if you can work directly on the tilt series, then in some ways it's like basically the same as single particle. You actually even get more information about the heterogeneity of a particular particle since you see it from many separate tilts. So if the upstream tools exist to extract that, to like track where the particles are within the tilts before you get to the subtomograms, then it would be very directly applicable, and you wouldn't have to deal with things like the missing wedge and other, I'm not actually too familiar with many of the other aspects of subtomogram averaging, but that is definitely something that we're interested in seeing if it can be applied there.
Great, thanks. Yang Jin Cho is asking, just curious if you can apply such CryoDRGN method, how much efficiently, onto RNA that has high heterogeneity as well.
Yeah, I don't see any reason why you couldn't apply it. I haven't myself worked on heterogeneous RNA data, but especially if you can get a consensus reconstruction, then it's reasonable. If there's enough structure to your RNA that you can do that, then that would be interesting. And I think, again, this motivates the ab initio approach for heterogeneous reconstruction.
Thank you. Pranav Shah is asking, can you explain how one would go about estimating heterogeneity for symmetric molecules? Presumably the analysis is complicated because of the symmetry?
Yeah, that's a great question. So I think, especially for these pseudosymmetric molecules where the underlying protomers are, you have different, I'm trying to go back here, where you have different copies of the same molecule, but maybe there's variations in the different copies. What I've been recommending is you just treat it as asymmetric. You just run a C1 refinement to get your poses, and then use CryoDRGN to fit the residual heterogeneity of the different subunits, the different protomers of your symmetric complex. One thing that I have noticed is that in the C1 refinements, it's important to start from an initial model that is asymmetric, because if your complex is mostly symmetric, so I bring up this slide since this is the RAG1, RAG2 complex, that's C2 symmetric in the core. In the DNA extensions, it's not C2 symmetric. We found that starting from an initial model that had these asymmetric extensions is really important to make sure the alignments were the particles were aligned to the correct symmetry copy. And then one thing that came up in the ab initio approach is that some of these lower resolution elements were much better resolved by treating the heterogeneity ab initio instead of fitting it to a C1 reconstruction. And maybe that's just because of some of the dynamic masking and the traditional algorithms that really focus in on the core. And in our approach, I'm just treating the whole image as the object to align. So maybe it's something because of that, but it's definitely the symmetry is definitely a challenge in making sure the poses are correct, but otherwise we haven't seen too many other issues.
Great. There's lots and lots of questions. Maybe I'll take one last question, and then you will have to answer all of them in the Q&A in the interest of time. Let's see. How well does this software work for membrane proteins in nanodiscs or detergent micelles where there may be heterogeneity in the membrane mimetics?
So I think we've gotten pretty good success for proteins and membrane for some of these membrane protein complexes. It is a lot more challenging because sometimes these membrane protein complexes are pretty small, and then the heterogeneity of the micelle dominates. One thing that's worked decently well is to do signal subtraction of the membrane of the micelle or the nanodisc. And maybe that's only possible if the micelle itself is relatively stable across the data set. And so that's something that we've kind of looked into, but haven't rigorously evaluated. But the idea is you definitely want to remove as much of the non-structural or the variability that you don't actually care about from the data set as much as possible.
Thank you so much, Helen. I think in the interest of time, we will stop here because we're already over time. I'd like to thank you again for a really beautiful talk, and you can see there's a lot of interest in the audience. And also, I want to thank again for all the speakers in this session and all the participants.